Let's start the basic topics in immunology. What is immunity? Types of immune response, that is, innate immunity and adaptive immunity, mechanism of innate immunity, phagocytosis, types of adaptive immunity, active, passive, humoral, and cell-mediated immunity. Immunity. The word immune comes from the Latin word immunes, which means exempt. So, what is immunity? Immunity is the ability of an individual to offer resistance against infection caused by pathogenic microorganisms. Mammalian immune system encompasses myriad of cells, tissues, and organs that work together to destroy the foreign invaders. Vaccinations render assistance to people by providing immunity to certain diseases caused by bacteria, viruses, and harmful substances. Immune response. There are two components of immune system. The first one is innate immunity, which is considered as general immunity, and another one is adaptive immunity, which is considered as specialized immunity. The image here depicts the difference between innate and adaptive immunity. Innate immunity fights against foreign antigens, injuries, and pathogens, whereas adaptive immunity fights against specific pathogens or changed body cells. The innate immune system prevents the entry of germs into the body by physical barriers such as skin and mucous membranes. Bacteria or viruses that enter the body can be stopped straight away by phagocytes, which are also known as scavenger cells. If the innate immune system fails to destroy the germs, the adaptive immune system takes charge. The adaptive immune system provides protection by B cells, T cells, and antibodies. This picture represents the components of innate immunity and adaptive immunity. In innate immunity, mononuclear phagocyte system comprises macrophage, dendritic cell, and monocyte. Other components include natural killer cell, complement protein, and phagocytic cells such as mast cell, basophil, eosinophil, and neutrophil. The cellular components of adaptive immunity are B cells, which produces antibodies, and secondly, T cells, which are of two main types, that is, helper T cells, CD4+, and cytotoxic T cells, CD8+. This antibody-mediated immunity is termed as humoral immunity, and T-cell-mediated immunity is termed as cellular immunity. Gamma-delta T cells are a small subset of T cells. Natural killer T cells share properties of both natural killer cells and T cells. Gamma-delta T cells and natural killer T cells fall in between these two categories. The host uses both innate and adaptive mechanisms which work closely together to detect and eliminate the pathogens. If the innate immune system fails to destroy the pathogens, the adaptive immune system takes charge. Innate immunity. It is also referred to as nonspecific immunity. It is considered as the body's first line of defense mechanism against all germs and foreign substances. All individuals by birth possess this natural resistance, which is why it is referred to as the natural immunity or innate immunity. Mechanism of innate immunity. It comprises 4 defensive barriers that keep harmful materials from entering your body, and they form the first line of defense mechanism in the immune response. It includes anatomical barriers, physiological barriers, phagocytic barriers, and inflammatory barriers. If an antigen crosses these barriers, it is attacked and destroyed by other parts of the immune system. Firstly, anatomical barriers, which include skin and mucous membrane. The outer layer of skin, called epidermis, is composed of keratinocytes and Langerhans cells, which provides protection to the skin. The middle layer of skin, called dermis, is composed of sweat glands that physically washes away the microbes, and sebaceous glands that produces oily secretion called sebum. The sebum is composed of lactic acid and fatty acid that maintains low pH of skin. This acidic pH inhibits the growth of most microorganisms. Conjunctivae, gastrointestinal tract, genitourinary tract, and respiratory tract are lined by mucous membranes. The viscous fluid mucus is secreted by the epithelial tissue, which entraps the foreign pathogens. The hair-like projections of epithelial tissue, called cilia, flushes out the pathogens, followed by coughing or sneezing. Secondly, physiological barriers. It includes high body temperature, low pH of sweat glands, sebaceous glands, and gastric juice, that is fatal to many pathogens. Chemical mediators such as lysozyme, interferons, and complement proteins. Lysozyme is a hydrolytic enzyme present in saliva and tears. It cleaves the peptidoglycan layer of the bacterial cell wall. Interferons are the antiviral proteins produced by the virus-infected cells. Complements are a group of 30 serum proteins that circulate in an inactive state, and forms the membrane attack complex to destroy the pathogenic bacteria. Thirdly, phagocytic barriers. Phagocytosis is defined as the process of engulfing the microorganisms by phagocytic cells, such as monocytes present in blood, macrophages present in tissue, neutrophils, dendritic cells, and B lymphocytes. These phagocytes internalize, kill, and digest the pathogen. This is a diagrammatic representation of phagocytosis mechanism. This is the plasma membrane of the phagocytic cell, and this cellular extension is called pseudopodium. Initially, the bacterium becomes attached to the receptors on the surface of the phagocytic cell. Subsequently, the bacterium is ingested by the phagocyte, thus forming a phagosome. This process of engulfing the pathogen is termed as phagocytosis. Phagosome then fuses with the lysosome, forming a phagolysosome, followed by degradation, where the lysosomal enzymes digest the pathogen, and forms residual bodies. Eventually, the digested products are eliminated from the cell by the process called exocytosis. Mechanism of phagocytosis. As discussed earlier, there are 4 sequential steps in phagocytosis. The initial step is entrapment, where the bacterium becomes attached to the pseudopodium of the phagocytic cell. The next step is formation of phagosome and phagolysosome. Bacterium is ingested by the phagocyte, thus forming a phagosome. This process of engulfing the pathogen is termed as phagocytosis. Phagosome then fuses with the lysosome, forming a phagolysosome, followed by degradation step, where the lysosomal enzymes digest the pathogen, and forms residual bodies. The final step is exocytosis. The digested products are eliminated from the cell by the process called exocytosis. Fourthly, inflammatory barriers. Inflammation is the hallmark of the innate immune response. Roman physician Celsus reported 4 signs of inflammation, which often leads to fatigue: rubor, tumor, calor, and dolor. Functio lisa is sometimes mentioned as a 5th symptom. The inflammatory reaction brings in phagocytic cells to the injured site to clear cellular debris and aids in wound repair. So, here you can see that, in case of an injury, harmful things enter the body, resulting in inflammation, which is characterized by redness, swelling, heat or warmth, and pain. This image shows the 4 major events in inflammatory response. First of all, in the event of tissue injury, any tissue damage caused by wound or infection releases the bacteria, which causes the resident phagocytic immune cells to secrete the potent inflammatory mediators such as histamine. This leads to vascular events, which comprises vasodilation and increased vascular permeability. Histamines are vasodilators that increases the diameter of local capillaries, causing an increase in blood flow. At the same time, histamines increase the permeability of local capillaries, leading to cellular events. There is an influx of cellular fluid called exudate and the cells into the surrounding tissues. The ultimate step is recruitment of phagocytes, where the neutrophils and other phagocytes from the blood are extravasated to the site of inflammation by chemotaxis. Neutrophils phagocytose the pathogens and destroy them. The 4 complex sequence of events that occurs during inflammation are detailed here. Tissue injury. Tissue damage caused by wound or infection releases the bacteria, which causes the resident phagocytic immune sentinel cells to secrete the potent inflammatory mediators such as histamines, leukotrienes, and prostaglandins. This leads to vascular events, which comprises vasodilation, characterized by heat and redness of the inflamed tissue. In this step, the vasodilators, like histamines, increase the diameter of the local capillaries, causing an increase in blood flow. Prostaglandins cause vasodilation, and is the major source of pain until the tissue is healed. Following this vasodilation step, there is an increased vascular permeability, characterized by edema. Histamines increase the permeability of local capillaries, leading to cellular events. There is an influx of exudate and cells into the surrounding tissues. The last step is recruitment of phagocytes. Leukotrienes attract the neutrophils and other phagocytes from the blood, which are then extravasated to the site of inflammation by chemotaxis. Neutrophils phagocytose the pathogens and destroy them. The accumulated cell debris is visible as pus at the infection site. This is another picture briefing the inflammation events. Any tissue injury caused by physical or chemical agents or pathogens brings about capillary widening, where there is an increased blood flow, responsible for heat. Later, the fluid releases into the tissues, generating redness and swelling. The leukocytes extravasate to the site of injury, causing tenderness. Finally, there is a systemic response in the form of fever, inducing pain. Let's illustrate the mechanism of fever. When pathogens invade the body, they activate the leukocytes in the body, which triggers the release of cytokines. These cytokines stimulate the hypothalamus to produce prostaglandins, which is the major source of fever and pain. Adaptive immunity. It is the immunity that an individual develops during an initial infection with exposure to a pathogen. It is also known as acquired immunity. It is highly specific and effective, as it builds a defense by producing antibodies against that specific antigen. The adaptive immune system comprises B cells, T cells, and antibodies. B cells and T cells are present in the tissue between the body's cells, whereas antibodies are present in the blood and other bodily fluids. Let's go through some of the important functions of adaptive immune system. The first one is specificity. It is extremely antigen-specific, and confers resistance by producing antibodies against a specific pathogen. The second one is diversity. It recognizes billions of different structures on an antigen, and effective at attacking a wide variety of pathogens. The next one is self and non-self recognition. Autoimmune response does not occur under normal conditions, but recognizes anything that is non-self, which are the foreign antigens. Lastly, immunologic memory. Memory cells that are formed upon the entry of antigen, reacts rapidly on subsequent exposure of the same antigen. Types of adaptive immunity. Adaptive immunity is classified on two bases. One is based on mode of development, and the other one is based on effector molecules. It is of two types. Active immunity and passive immunity, which is further classified into natural active immunity, artificial active immunity, and natural passive immunity, artificial passive immunity. Now based on effector molecules, they are categorized into two types, which is humoral immunity, also called antibody-mediated immunity, and cell-mediated immunity, also called T-cell-mediated immunity. So, what is active immunity and passive immunity? In active immunity, the host develops an adaptive immunological response by producing antibodies upon exposure to a disease organism. It is long-lasting, and can sometimes persist lifelong in the host. In passive immunity, the host receives preformed antibodies to a disease rather than producing antibodies through their own immune system. It is typically short-lived, and usually persists only a few weeks or months. In natural active immunity, the immune system responds by producing antibodies against the pathogen that can cause infectious disease. Examples include measles, chickenpox, where immunity lasts lifelong, and influenza, where immunity lasts only for few years. Artificial active immunity is also known as vaccine-induced immunity, as it is acquired through the introduction of a killed or weakened form of the pathogen through vaccination. Examples include live attenuated vaccines such as BCG vaccine that prevents tuberculosis, and Sabin vaccine or oral poliovirus vaccine that prevents polio. Another type of vaccines are killed or inactivated vaccines. Hepatitis A vaccine that protects against hepatitis A virus, and Salk vaccine that prevents polio. In natural passive immunity, antibodies are transferred from one host to another. For example, infants acquire passive immunity from its mother because they are born with maternal antibodies that are transferred through the placenta during pregnancy. These antibodies usually disappear between ages 6 and 12 months. In artificial passive immunity, antibodies are produced outside the host and are transferred from donor to recipient. It provides immediate protection from a specific disease. For example, patient is injected with convalescent serum to boost the survival rate. Immune serum globulin is given for hepatitis exposure. Humoral immunity and cell-mediated immunity are the two types of an adaptive immune response that enable the human body to defend itself in a targeted way against harmful agents such as bacteria, viruses, and toxins. Humoral immunity. Humoral response is primarily driven by B-cell lymphocytes that produces antibodies against foreign antigens, which are freely floating in the bloodstream or outside the infected cells. Hence, it is also known as antibody-mediated immunity. It is so named because humor refers to the body fluids, generally blood, where the antibodies circulate freely and bind to the specific antigens, neutralize them, and causes lysis or phagocytosis. The second type is cell-mediated immunity. Cellular immunity occurs inside the infected cells, and is mainly mediated by T-cell lymphocytes of the immune system. Hence, it is also known as cell-mediated immunity. It involves the activation of antigen-specific cells, such as cytotoxic T lymphocytes and macrophages, which recognizes and destroys the infected cells harboring antigen. It primarily defends against intracellular pathogens, multicellular parasites, transplanted tissue, and cancer cells. T helper cells and cytotoxic T cells are the key effectors of this cellular response. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.